maybe don't know me, I am not a song leader, so help me out. 218. Men especially will need you to bellow it out, okay? I know that my Redeemer lives in every praise for me. I know eternal life He gives, from sin and sorrow free. I know that my Redeemer lives. I know eternal life He gives. I know that my Redeemer lives. I know that over yonder stands a place prepared for me. A home, a house not made with hands, most wonderful to see. I know that my Redeemer lives. I know eternal life He gives. I know that my Redeemer lives. My Redeemer lives. Let's pray. Heavenly Father, thank you so much for this chance to be here this morning and gather together and be encouraged by the evidence that there is a Creator, that you are Him, and that we can put our full trust in you. The things that we see through this world can be made known about you. Thank you so much for being such a good God, and thank you most of all for Jesus, and it's in His name we pray. Amen.
Thank you, Josh. Good morning. It's good to see all y'all this morning. Y'all came back. I'm really happy. That's really good. That's great. Josh, I didn't change the arrow on there, but is that okay? I think we can see it. Yeah, I think we can see it. Can y'all see that arrow?
Yes.
I don't see it at all. Oh, it helps if I turn it on. All right. Now do you see it? Yeah, there we go. All right. There we go. All right. Let's get started. We don't have much time. Oh, what's my time? When do I have to?
10 o'clock.
10 o'clock. Okay, we'll make it work. All right,
10
very
or 5
good.
if you want.
Very good. We're always concerned about how close are we to chimpanzees. I know there was this one family, and the little girl went into Dad and said, "Dad, where did we come from?" He said, "Well, long time ago, there was this ape-like thing, and we kind of switched over, and we became humans from these ape people." And so she went to her mom and said, "Mom, where did we come from?" And she said, "Oh, God made us and created us," all like that. And the girl said, "Well, that's not what Dad said. He said we came from monkeys." And the mom said, "Well, that's his family." Yes, that's pretty much. That's probably a bad joke. Yeah, there we go. Tracing our DNA back to Adam and Eve. It's been fun to be in the biology world and to see what all is going on in that. And now that we can do genetic sequencing and the genetic technology, we're finding out a whole lot about the whole creation, not only humans, but all creation. And it's just been great because it's just magnified how complex and how amazing God is and what He has done in all of this. Here's what we've heard. We've heard this for years. And this is a quote by Ann Gibbons. She is an evolutionist. Back in 2012, she said this: "Ever since researchers sequenced the chimp genome in 2005, they have known that humans share about 99% of our DNA with chimpanzees, making them our closest living relatives." So that's been the idea through science all along. We're about 99% like a chimpanzee. Actually, it's 98.5, the part that they're looking at. But a lot of this is based on the 1970s data. They took some human and chimp DNA, mixed it together, heated it, cooled it down so that the DNA would align with each other, and then they looked at the cloudiness of it and determined, well, we're about 98, 99% similar. And so just by how cloudy the mixture was, that's how they determined that. It wasn't very accurate at all. And they only measured the DNA that would align. Now we know that humans have these large sections of DNA that chimpanzees don't, and chimpanzees has these large sections that humans don't have. So it's kind of interesting how it's kind of evolved over time, the thinking of going on, of what's going on with that. After they sequenced the genome, then we heard this from the director of the NIH, Francis Collins, even promoted this. If you look at our DNA, we're 99% the same. But they're only looking at the DNA that codes for proteins. They don't tell you that. And that's only 2% of our DNA. We've got all this DNA, and they think it's only the proteins that matter, but that's only 2% of our DNA, and that's all they're looking at to make that comparison. And at that particular time that they sequenced the DNA, they thought half of our DNA was junk. So, well, if we're 98, 99% the same as chimpanzee, what else they don't tell you is humans and mice. We share 97.5% of our DNA that codes for proteins. So are we closer to the mouse? Are we closer to the chimpanzee? We're probably the same distance to a horse on our proteins. We all have about the same proteins. We live in the same environment. It's just going to be that way. And in 2020, these evolutionists, Seaman and Bugs, they reported a 96.6 similarity, showing we're really close to each other. But what they don't tell you in that, when you look at it, is they didn't count the centromeres, the telomeres, all the copy number variations in the DNA, about 300,000 small insertions and deletions, which is about 2 million letters out of the 3 billion letters, and an additional percent that resisted to align together. So a lot of times all we hear is the 96.6 or the 98%. They don't tell you the whole story of what they're looking at. So here's what's happened. The human genome was sequenced in 2003. It really wasn't completed until about 2 years ago. When they finished in 2003, they haven't done the Y chromosome, they haven't done centromere regions, they hadn't done telomere regions, and so it really wasn't completed until about 2 years ago. They finished the chimpanzee genome in 2005. So science was thinking this, that only the DNA that codes for proteins, that's all that matters, because everything in our body is run by proteins. And half the human DNA was junk, has no function at all. It's just left over from our evolutionary past. That was the thinking in 2005. That's only 20 years ago. So, the director of the NIH, he appointed 30 scientists to a project called ENCODE. And you can see it up there. That stands for Encyclopedia of DNA Elements. So, their assignment was, what proportion of our DNA has a function? What proportion has a function? That was 2005. In 2012, the people who were running the ENCODE project, these scientists, they held a press conference because what they had found was totally against everything that we had known. They said, our DNA is 100% functional. Every bit of it has a function. That's amazing. Which I'm not surprised. Are you surprised at that? God made us to where he didn't put junk in our DNA. Every bit of it has a function. That's a problem for evolution because they need that junk DNA as a place for mutations to occur because we do mutate. We do have a lot of mutations. We each one have about 100 more mutations than our parents. So we've got a lot of mutations going on. Most of them are neutral, but we have mutations going on. They need that area for the mutations to occur in so that it doesn't mess up our DNA. Good morning, you are late for class. These guys are in biology class over at Harding. I know 3 of them are, yeah. Are y'all in biology class? No, she's not. Okay, there we go. Now then, so no junk DNA. Every bit of our DNA is functional, which is really, really kind of interesting and good to know. So, since things were sequenced, they started comparing humans and chimpanzees. And there's 5 studies that they looked at, and you can see they went from 80 to 90% similar to 84% similar to 89 to 90 to 85% similar. And the problem with all of this was to sequence the chimp genome, they used the human genome as a scaffold. So they would use the human genome and they'd find this piece of chimp genome and it matches up to this one in the humans and this one matches up to this one. Well, when you do that, it kind of makes the two genomes really, really similar together and about the same. So that was kind of a problem. And also, when you're dealing with that, you have your own DNA on your fingers and things, and there's a lot of contamination, but you don't realize that the contamination is there. And so the early studies had a lot of contamination. They started figuring all that out and started reducing the contamination as that went along. So Jeffrey Tompkins, he is with Institute of Creation Research. He's a good researcher. He directed a genome center in the Department of Genetics and Biochemistry at Clemson University, so he knew what he was doing. He looked at 101 data sets that went into originally building the chimpanzee genome. He sampled 25,000 sequences at random, and half of them were extremely unlike human. The other half were extremely similar to human. And so they were probably contaminated with human DNA. So he, using the data that appeared to have much less human DNA, he compared the two, and he concluded we're about 81 to 85% similar to a chimpanzee. And so that's what he came up with in his calculations, which is a good calculation, and was on, and it's probably correct. So scientists have been realizing the contamination part, and so they've been working to eliminate that. So we're not as close to the chimpanzee as reported. We're no longer 98.5% the same. And that 15 to 20% is mainly large segments. So we have a bunch of large segments that you can't find in chimpanzee. You only find those in human, these large segments. And these large segments in chimpanzee that are just chimpanzee, they're nothing else. So that's really kind of good to find out. So we also need to ask the question, which never comes up, how close are we to other animals? So we're probably about 80% the same as a mouse. We do most of our studies with mice because their physiology is almost exactly the same as ours. And so we're about 80% the same as a mouse. A horse, we're really close to a horse in our DNA, things like that. So being 81 to 85% the same as a chimpanzee is not really a big deal because we're about that same percent with other animals as well. So it's interesting. We share 75% of our genes that cause disease with single-cell bacteria. Is that not amazing? That's just amazing. So we have lots of similarities among all organisms. God made us that way. We all live in the same environment, so we have to have the same DNA to be able to look at it. So here's my point. Evolution tries to present things as really simple and easy to understand so that you can go ahead and believe it. They don't show us all the information. They'll tell us about humans and chimps are real close together, but they won't tell you you're real close to a mouse. They just won't tell you that. So they leave out a lot of pertinent information as well that's kind of important to all that. So, for instance, let's look at this. This is in our textbooks. This is showing human evolution, and y'all can kind of see they think we diverged over here 6 million years ago. This is 6 million, 5 million, 4 million, 3 million, 2 million, 1 million years ago. So somewhere around 6.5 million years ago, we diverged. And this is our path through Australopithecines and then to Homo over here. And so the green bars kind of tell you the time where they find fossils of all of those. And so that's what they show in the textbooks on what's going on. Now then, I want to draw a line. Can I draw a line? Yeah, I'm going to draw a line right there. If you draw a line right there, what's on the right side of that line? That's all Homo. I know it's Homo habilis, not Homo habilis, but Homo ergaster, Homo sapien, Homo neanderthal. All of the homos, that's all human on that side of the line. If you look on the other side of the line, the Australopithecine line, all of those are really, really small. There is a Homo over here. There it is, Homo habilis. They have only found 2 fossils that give any indication of its height, and it's only 3 feet high, and it's mainly Australopithecine. In fact, when I was in school, it was called Australopithecine habilis. and now they moved it to Homo, then they moved it back to Australopithecine. It's really a hodgepodge of bones. They really can't decide whether or not it goes in the Homo section or Australopithecine, but it's definitely Homo Australopithecine. And the reason I show you that is because if you look at what's on this side of the line versus what's on the other side of the line, that's the difference. That's the difference. So you can tell, they only show the skulls when they're showing this, but there's huge differences between the Homo and the Australopithecine. This is from the Journal of Molecular Biology and Evolution. No gradual series of changes in earlier Australopithecine population clearly leads to the new species, early Homo, and no Australopithecine species is obviously transitional. So if you look at the research that's going on, there's not very many similarities between the two sides. They're just quite different from each other. Australopithecines, that word actually means southern ape. The brain and the body are about the size of a chimpanzee. The most famous is Lucy, and we always hear about the fossil Lucy that was found that started all of this. Australopithecines, they are ape-like in all of those structures you can see on the right. Not human-like at all. And their arms, their hand bones, their striding gait, their shoulders, their toes, their brain size, their teeth, their chest, inner ear canals. There's just no similarities at all to humans. They're strictly Australopithecine. So Lucy, she was very small-brained, 300 cubic centimeters in her brain. Humans, our variation, we go from 500 cubic centimeters to 1,200, 1,500 cubic centimeters brain size. Some of us have big heads, some of us have small heads. But Lucy was quite a bit smaller than that. Bipedal? No, she was not bipedal. Nature, the article in Nature, the hand bones showed Lucy knuckle-walked, just like chimps and gorillas. They're very suitable for climbing trees. Their arm show that. The pelvis was badly crushed with distortion. There was error in reconstruction, creating a very human-like shape. And so there's such a desire in that paleoanthropology world to find the closest relative to human. There's such a desire that they make things look like humans, whether or not they do or not. So it's kind of interesting. University College in London, anthropologist Leslie Ayala said this: Australopithecines are like apes, and the Homo group are like humans. Something major occurred when Homo evolved, and it wasn't just in the brain. So there's obvious differences to the scientists who are working in this area that shows there's just lots of differences. This is what is in our current textbook that we use at LCU. Which book do y'all use? Who's the author, do you know? No? Okay, all right, that's all right. That's okay. I know that's not important. No, there we go. So that's what it looks like now. Instead of just the skulls, they're showing actual whole figures there to look at the difference. So you know what I had to do. I had to draw my line. I draw my line right there. There's the line. On one side of the line, all of those are 6 feet tall. On the other side of the line, they're all 3 feet tall. One's 4 feet tall, but he's more ape-like than all the rest of them. So there's still a big difference. Now, if I go back, can y'all really tell there's a difference in size? No, they don't really want you to see the differences in all of that. But it's quite clear if you look at all the fossils that all of these are 6 feet tall, all of these are 3 feet tall. They're Australopithecine versus Homo. It's just a big difference in all of those. And so I wish they'd add more stuff to their charts, but they're not going to do that as we go along. Real recent finds, Ida was found in 2009. Turned out to be just a lemur, a very extinct animal. There was a lot of hype on the media about that. Lucy was just an Australopithecine. Homo habilis is just a hodgepodge of bones, very unclear where it should be. Naledi in 2015 was found. It's just an Australopithecine, a very recent Australopithecine, in fact, is what it turns out to be. Java Man, Nebraska Man, Piltdown Man, they were all hoaxes. I think with Piltdown Man, 500 doctoral dissertations was done with that. Turned out to be a hoax. Yeah, that's kind of sad. Neanderthal, all human, 99.7% of our DNA is exactly the same. The Neanderthals lived in the upper part of Europe, kind of in the colder region, and they were kind of separated off to themselves a lot. A lot of inbreeding and within families went in with the Neanderthals, but they were a really kind people. And if you look at their culture, what they found in paleoanthropology is they're really a kind people and cooked and did everything. So they're human. They were just human. They just lived in a different part of the country. Homo erectus, Homo ergaster, Homo heidelbergensis, they're all human. Those are just all human. There's no clear evidence of a connection between humans and chimps of the great apes. You just can't find it in the literature. A major review was done on all the hominids. So humans and the great apes, we call those the hominins. The evolutionary sequence for the majority of the hominin lineages, apes, chimps, humans, is unknown. Most hominin taxa, particularly early hominins, have no obvious ancestors, and in most cases, ancestor descent sequences cannot be reliably constructed. So that was a major finding from a group that really tried to look at it objectively. And so that was their conclusion, which I thought was really, really good. Then this came out this year. It came out last month. It was just kind of fun. And we've been waiting for this to happen. It actually came out in the evolution world. It was an evolutionist. Wu is the main author of this study. They looked at the chimpanzee genome by itself. They didn't compare it to the human. They actually did a sequence of its genome by itself. In fact, they did all of the great apes that way to see how close we are together. And so they did a great study. They did it according to high standards. They did the proper filtering protocols. They did everything that they were supposed to do in comparing all of the great apes to see how close we are together. So this is what they found. There we go. So the way we need to read this, and I need to point that out up here. So orange, the orange color is within the species. So in the human species, among all the humans that they sequenced their genome, there was a difference. So we were about 3% different. So in this room, all of us, we could be about 3% different in our DNA. We just have different things going on and just a lot of differences that way. You will see that the dark blue is when you compare human to the ape. So comparing human to chimp, we are 15.2% different. So that seems to be pretty accurate. That seems to be where we're going to land with all this. We're 15% different. Now, if you go the light blue, it's going ape to human. So when you compare ape to human, it's not quite 15%. And so it's a little difference when you compare this one with this one rather than this one to this one going the other way. You can get some differences there. And so, yeah, we're about 15% different from chimpanzee. We're also about 15% different from bonobo, which was kind of interesting. Nobody ever thought about that one. From the gorilla, we're quite a bit different. And the orangutan, we're quite a bit different, 20, 25% different. So this is now official. These were evolutionists that did this study, and these studies were conducted in a proper way. So this is going to be the new thing. We're now 15% different. So not the 98 to 99% that we've heard earlier. The data is not highly advertised, nor is the data readily noticeable in the paper. The paper's 100 and something pages long, and it's kind of hard to find all the data and all that. I'm going to go back. Robert Carter with Creation Ministries International, I was reading his article on that. He is a geneticist with Creation Ministries International. He's a marine biologist, was at the University of Florida, but now he's working for Creation Ministries. He's a really good geneticist. I follow him on genetics on everything. He's just a good person to follow. And so I'm reading his article in reviewing all of this. And so he's the one that said it was hard to find all this material in this particular paper. So we should expect a high similarity with chimpanzee. We have the same body plan. We're both upright, and we have 4 limbs, 2 legs, 2 arms. Physiologically, we're really similar. Behavioral, we're real similar. So you would expect our DNA to be pretty similar. But there's that 15% that's just human. And that 15% is just chimpanzee. And that's a lot. That's a lot of our DNA. But it specifies us. Because we live in the same environments, you know we're going to have the same DNA. And I'll show you a comparison here in just a minute with other organisms. So why is this such a big deal? Well, the parts of the genome that align are 98% similar. But when you add the segments that do not align, we're only 85% similar. So the question is, how many mutations occurred to give that difference? That's going to require a little bit more study in all this. But the problem with this for evolution is this. And I don't know if y'all can follow the calculations. I think you can. So let's go through these calculations real quick. If we have 100 mutations per generation, which is what we have, we usually have every generation about 100 mutations different from the parents. That means 50 mutations per haploid generation, like for mom or for dad. So 50 mom, 50 dad. And then if we diverge from a common ancestor 6 million years ago, so 6 million years ago, and if we consider a generation time of 30 years, we're kind of being generous there, or maybe not, then that would calculate for the 6 million years, we'd have 300,000 generations from our common ancestor. So if you got 300,000 generations, you multiply that by 50 mutations per haploid generation, that would give 15 million differences between each species and the ancestor. Or, considering both species, that would be 30 million differences between us and them today. So if you divide those 30 million differences by our DNA size, which is 3 billion letters, it comes up to a 1% difference. And so that's important for evolution. It's important for us to be only 1% difference because they think our mutation rate and our fixation rate, the rate that a mutation is fixed into the population as a whole, if it's 1%, that fits all the data, if it's 1%. So they've been wanting us to stay right at 98, 99% the same as chimpanzee because it kind of fits the mutation rate that we've got going on through time and all the generations that would have been covered through there. But now, we're 15% different. So this is a big problem for evolution. They don't know what to do with this. Because the 1% kind of worked with our mutation rate and fixation rate just fine. But now it's 15%, it doesn't work. So the simple thing to do was, we'll move our common ancestor back 15 times. Instead of 6 million years, 15 times 6 million years, that puts us to where our common ancestor is the time of the dinosaurs. And paleoanthropology is not going to let us do that. So they've got a problem. They've got a problem. So it's kind of fun. It's kind of fun to see what's going on with that. I don't know what they're going to do with this, but that's what's going on in that world. They're stuck, really. They're stuck. Yeah?
Has a built-in resistance to mutation. Mutations don't tend to be inherent. Oftentimes, in reproduction, the genetic sequence erases mutations. One organism can have mutations, but passing on, it is not common at all. It is actually very resilient to it.
Yeah, our DNA is resilient to it, but overall, though, we have a lot of mutations that are hard to detect because they're neutral mutations. We may change the third base, and that doesn't change the amino acid. So there's a lot of mutations that we just can't detect in there at all. But it's pretty close to about 100 mutations, yeah. So we're mutants, I guess, because we mutate a little bit. I wish I was, what's the guy on mute? Wolverine, yeah. I'd kind of like to be Wolverine or something like that. Okay, so what's ahead for this? Yeah.
Well, you would think that we would all have a similarity since we were all created from dust anyway.
That's right. That's exactly right. Yeah, and God's going to put the same DNA for a certain process. We all eat, so we have to have the same digestive system, the same digestive enzymes, and He had to do that for us to be able to be doctors and figure things out. Nobody wants to volunteer to be worked on, but we can work on animals. So yeah, it all works. Yeah.
Dr. Martin,
Yeah.
just for simplicity purposes, because I'm a simple man, there's a lot of gaps between what we know about the apes and humans.
Yes.
There's no real connection in between. The gaps are so wide in between.
Yeah, that's a good point. There are big gaps there that kind of separate us a lot. Also, and that brings up another thing, they may have a gene for this particular protein, and we have that gene for that particular protein, but we use that gene a lot. They don't use it. The gene could be there and be in our DNA, but not utilized at all. And there's several proteins that are like that. So there's lots of differences there that work in there. And only 2% of our DNA is the protein part. The rest of our DNA regulates those proteins. And so they're regulated completely different. So that's a good point to make is even though our DNA may say we're 85% the same, how it operates is not even close to 85% the same. Yeah, good point. I should have said that. Yeah. All right. So they're going to do something. I think they're going to do something to now that it's 15%, they're going to start saying, well, maybe we had huge mutations where large sections of chromosomes were affected. But the problem with that is our DNA is so intertwined, and we have so many genes affecting other genes that it's hard to change big sections without harming the individual really greatly. So I don't know where they're going to land. They'll land somewhere. It's not going to go away. So, oh well, we're in there. We're in there. Right now, we're 15% difference, and so that's better than the 1% that we've been hearing for a long, long time. So, this is an interesting article. I wanted to show you this. I even titled this thing, Tracing Back Our DNA to Adam and Eve. This is a study done, and I don't know if you can see their names up here. Carter, there's Robert Carter. He's a really good geneticist. But also J.C. Sanford. The middle guy, I don't know, but he's a mathematician. He had to do the statistics. So Sanford is probably the most well-known population geneticist in the world. He developed the gene gun for plant engineering. He's been at Cornell University for all these years. He retired a few years ago. When he retired, he wrote a book. And the book said, we as population geneticists have been looking for evidence for evolution for years, and we can't find any. We just can't find it. And he just told the whole story about what was going on. And visiting with him on the phone, he said that all of his colleagues had not contacted him after he wrote the book. They didn't call him up and say, you are wrong on this. Remember this study? Remember this study? They can't do that. Everything he said was correct. And so it's kind of nice to have professors do that stand for what they believe and all that. Anyway, he's a good geneticist. So they looked at the human Y chromosome. So only the males have the Y chromosome. It's passed from dad to son. And the human mitochondrial chromosome, which is only passed from mom to her offspring. So just to give you an idea of where these chromosomes are at. So here's a cell. Does this look familiar, guys? Yeah, it does. All right, all right. This up here, your chromosomes are in the nucleus of the cell. And so there's where all 46 of your chromosomes are at. The Y chromosome's in the nucleus up here as well. But over here in the mitochondria, it actually has its own DNA. It's got a single chromosome in that mitochondria. And so the egg, when the egg and sperm unite together, the sperm, all it's got is a nucleus with DNA. But the egg has all the cytoplasm with the chromosome, or with all the mitochondria in it. So mom passes the mitochondria to her offspring. So they were looking at those two chromosomes. And so They used the 1,000 Genome Project. This is a project that was done by secular biologists. They looked at 2,500 individuals from different populations all over the world. So they compared the genome of this tribe with this tribe right next to it, and from this tribe with the next country next to it. So they were comparing these people all the way around the world, different genomes, trying to compare their Y chromosomes. And that's what they came up with. And you can see everybody is connected together. All these letters and numbers are different tribes or different countries or regions in the world, and they're all connected together. Is that not interesting? So, and they could even know how the mutations occurred over time and could trace all those mutations back to the original Y chromosome of either Adam or Noah. We're not really sure. So, isn't that interesting? I just think this is really kind of interesting. They also looked at the Simmons Genome Diversity Project. It has a much wider range of people that they looked at and almost came up with the same connections among all those tribes. So both of those projects were given the same data for the Y chromosome. Then they looked at the mitochondrial chromosome that's passed by the mom. They also came up with connections, and everybody all over the world was connected, and possibly the root here, Eve, is what they're calling it, mitochondrial Eve, was right here. So, and they can figure out what mutations occurred over the generations to get us where we are today. Isn't that interesting? So they actually know the actual sequence of the original Y chromosome and mitochondrial chromosome, which is kind of fun. Now, here's what they said. A pattern emerged in that multiple major branches have arisen from surprisingly closely related individuals in a very short window of time. In both trees, multiple major branches can be traced to identical ancestor individuals that could be brothers or sisters or cousins. And so if you look at the Y chromosome, do y'all see a starburst right here? You see another starburst here, and you see another starburst here. The three sons of Noah. Isn't that interesting? They even say it down here at the bottom. I don't know if you can read it. They say that's probably the three sons of Noah that they can trace the Y chromosome back to. If you look at the mitochondrial chromosome, same three. You see a starburst right there. You see another one there, another one there. And that's the three wives of the sons of Noah. They can trace all that back just by looking at all the different countries around the world. Is that not cool? It's just kind of cool that they can do all of that. Here's what they concluded. We can know with a very high degree of confidence the actual sequences of the historical individuals that gave rise to each branch in both family trees. Given a few reasonable assumptions, we can estimate the time to Y chromosome Adam, Noah, and mitochondrial Eve. Both individuals lived less than 10,000 years. They know the mutations. They figured out the mutations. They know the mutation rate today, how often a mutation occurs in the Y chromosome or how often a mutation rate occurs in mitochondria. And using that rate and looking at the number of mutations that they found, less than 10,000 years. That fits the Bible. That fits the Bible. Isn't that great? Yeah, it's just fun. It's just fun. And so I just love this study. It's just really, really good. This article appeared in the journal Human Evolution. So this was a human evolution article, is what this one is. Fox News had an article about it on Sunday, November 25th. I know it was a Sunday because I was in church and my phone kept dinging. My students were texting me, have you seen Fox News today? Do y'all text y'all's professors very much? Good, I'm glad y'all don't do that. Yeah. And they said, you got to look at Fox News. So I went and looked at Fox News. So Fox News interviewed these two guys. They're the author of this article on human evolution, Stockley and Thaler. So here's what they found. And I hit the wrong button. Sorry, didn't mean to do that. All humans alive today are the offspring of a common father and mother, an Adam and Eve, who walked the planet 100,000 to 200,000 years ago, which by evolutionary standards is like yesterday. Can you believe that? This is a human evolution journal. Moreover, the same is true of 9 out of every 10 animal species came from a common father and mother. Is this sounding familiar to you guys? Took a male and female on the ark? Yeah. And so, meaning that nearly all of Earth's creatures living today sprang into being recently from some seminal Big Bang-like event. So they got a little evolution thrown in there just a little bit, but that's okay. They were looking at the mitochondrial DNA. They looked at 5 million animals, sequenced their mitochondria from 100,000 different species to come up with this data. And that's what they came up with. All these species came from a common father and mother. Isn't that great? Reminds you of the ark. Thaler, one of the authors, said this: "This conclusion is very surprising, and I fought against it as hard as I could." He did not want to come up with that data, but he came up with that data. So that's funny. That said, Stocklin-Thaler's study does line up with the Bible in at least 2 notable ways. First, it affirms that we and our fellow creatures on earth arose from a recent and profound creation event orchestrated by some unknown mechanism. You and I know that mechanism. And second, the DNA barcodes reveal that species are quantized. They fall into very distinct, widely separated populations, what the Bible calls kinds. And so we learned yesterday when we talk about kind, we're talking about a family, the dog family, the cat family, something along those lines. So that's interesting, isn't it? This is evolution people talking. So I went to the original article on human evolution to get their data, and it was surprising to me. It really was surprising to me. You'll look on this axis of this graph here. Here's the y-axis here. This is the average genetic difference within a species. And look at all the species. 100,000 species. They're all down here about 0.1%. About 0.1%. I usually, sorry, I bump this every once in a while. Really tall. A small amount of difference within a species. We just haven't very, very much at all over time. And all the species are like that. Here's humans. We only vary 0.1% is all we vary. That's all we vary among humans. And all of those are like that. Now, if we diverged from chimpanzees 6 million years ago, then that wouldn't be right there. It would be way up here on a large percent because of our mutation. But we're just not different, which means we haven't been around that long. Is that not cool? It's just really, really cool. I just think that's really funny. If you go to this PhysOrg website, they review papers a lot on that website, and they were saying on that website, genetically, the world is not a blurry place. It's hard to find intermediates, the evolutionary stepping stones between species. The intermediates disappear. Isn't that fun? Yeah, just really, really fun. So, the amount of research for intelligent design is growing. Most of the data fits the biblical model for creating. The increase in research is bringing a greater clarity to origins, and it's favoring the biblical model of creation. So, again, I get a lot of my data from scienceandculture.com. They used to call it Evolution News, but it's really good. That's an intelligent design group. Creation.com is a really good resource. That group is Creation Ministries International. My favorite geneticist is at that place, Robert Carter, and I follow him on everything. ICR has been pretty good here recently, and I like what they're doing. Answers in Genesis has a few research projects going on that are really good as well, and you can check those out. Dave Reeves Ministries down in Nashville is great. He's within the church and does a great job of keeping up with all this. And so I follow him a lot and what he comes up with. Last thing I want to show you is this dependency graph. Winston Ewart decided to start looking at the DNA, and is there a better way that we can classify organisms rather than the evolutionary tree? So here's the evolutionary tree. Everybody knows the evolutionary tree there. There's the tree for animals. We have a common ancestor. We have a common ancestor way down here. And it's funny, they say we have a common ancestor down here. They show everything up here that's living today. I can't do this left-handed, I do it right-handed. But they can't fill in the blanks down here. They never can fill in the blanks on who the common ancestor is down here, which I think is quite interesting. Anyway, they, if you use the phylogenetic tree, that was the tree of life there, the first representatives of most major groups appear suddenly, and there's no intermediates preceding them. And so we looked at that yesterday at the fossil record. All along the fossil record, things appear in the fossil record, but they can't connect it to anything below there. There's no precursor. And so that's always been a problem for the fossil record, and that's why they don't show you a complete fossil record with all the organisms in there, because they can't connect them together. And the pattern of physical traits and molecular sequences in species do not imply a consistent evolutionary tree. For instance, an octopus has an eye just almost exactly like our eye, but the octopus is way over here on this part of the tree, and humans are way over here on this part of the tree, and the way you do things in biology, if they're real similar in something, you put them really close together. We saw that yesterday. The hippopotamus and the whale are right together because the hippopotamus spends some time in water, so does a whale, so they put them together on the evolutionary tree. And so that's a problem for them. And groups of species have at best only 35% the same characteristics. So to put two of them together, they only have to be 35% the same. That means 2/3 of the data doesn't fit. But that's how they do it. If it's only 35%, they'll put them together. They'll say they're really close on the tree. So there's some problems with the tree of life that Darwin first started. He's the one that drew the first tree of life, and we've kind of followed that all along in biology. So Winston Ewart, in this particular journal article, he said this, and it's written there across the bottom. This hypothesis, the dependency graph of life, postulates that different biological species share modules related by a dependency graph. So he said, why can't we look at the DNA and see how close we are together among all the species? That's called a dependency graph. So let's take a look at what his reasoning is. At the top of this slide, at the top, that's a tree of life thinking. So we had a common ancestor a long time ago here for mammals, and then we had this one branched off and this one branched off. This is over in Laurasia. This one is over in Africa. And so they branched into two different parts of the country. And from that, we get the elephant, the manatee, the seal, the dog, the dolphin, the cow, and the bat. And so that's what a tree looks like, an evolutionary tree. Well, the problem with this is, if you look down here, the bat and the dolphin, they're two different parts of the tree, but they both have echolocation. And that echolocation is almost exactly the same. It's about the same proteins and everything. How can this group over here evolve something that's exactly like this group over here? That becomes a problem. And then you can also see these guys over here, the dolphin right here, and the seal and the manatee, they're all marine animals. They have to live in the ocean. And so their bodies have to accommodate the ocean characteristics, but they're on different parts of the tree. And so there's a lot of things that don't line up, is what he's saying with all of this. So he analyzed the DNA. And this is a dependency graph. And it's kind of hard to read, so let me work you through it. So he looked at the mouse. That's this color right here. I don't know what color that is. I call it puke, but I don't think that's a very good color to say. That's pink or something. This blue coloration, that's the zebrafish. And this green over here is the chicken. So he compared the DNA, a chicken, a bird, a fish in the water, and a mouse, a mammal. He compared these three, looking at their DNA. So if it's, look up here at the size of the circles. That indicates the number of gene families that they're looking at. So what is a gene family? A gene family is, well, in our body, we've got the hemoglobin family. We've got 5 different types of hemoglobin in our body. So 3 of them work while we're in the embryo stage, and then the alpha and beta hemoglobins work while we're in the adult stage. And then we got myoglobin, which works in our muscles. So I guess we got 6. So we call that a family. That's a gene family. 6 different types of hemoglobin that's in our body. So those are gene families. So the bigger the circle, the greater the number of gene families. And so if it's all green, you only find that in chicken. If it's all blue, you only find that in zebrafish. If it's all pink, you only find that in the mouse. But look at this. These gray, you find that in the mouse and in the chicken, in the gray. But look at this. All 3 of them share all of these gene families. We share tons of information with the fish and the chicken. Isn't that interesting? Yeah. And so he said, why don't we set up the tree of life according to this, according to genetics, and call it a dependency graph. And it kind of gets you to realize God used the same DNA in all organisms, but this 15% is just human. It's just human, only found in human. Does that make sense? Yeah. So he did the statistics, and he compared the tree of life to a dependency graph, and he used a Bayesville, log Bayes kind of statistics. He looked at 9 databases of gene families, and I know I've got 1 minute. I'll be done in 1 minute. So he compared the tree of life to no tree of life, and a tree of life was better than not having a tree of life. Then he compared a dependency graph with no dependency graph, and it was a whole lot better than no dependency graph. But when he compared the dependency graph with the tree of life, the one on top is the one that you say it's better. So the dependency graph was 100,000 times better, 500,000 times better. I can't read those numbers back there. 80,000 times better. The dependency graph was a much better way to classify organisms. So we're hoping one of these days we're going to switch to dependency graphs, get rid of that tree of life. Darwin was the one who got that tree of life stuck in our minds. He's the one that started all that. And that was 1859? Yeah. And so hopefully biology can switch over to looking at DNA and comparing organisms that way. And then we can really see that maybe people can see that God used the same DNA in all organisms. We just are special in certain areas that makes us special that way. Does that make sense? Yeah. So it's been fun to be in biology to see this happening and everything with all of that. I'm done. Okay. Questions? I don't know if I'm allowed to do questions. Yeah. Come ask me afterwards. That'll be just fine. Yeah.